Okay, hi everyone. Welcome to our 8 Notes 2023 lecture series. I hope you guys are all doing really well and having a wonderful term break. And I hope you've had a wonderful term 2 of year 12 as well. So once again, a very warm welcome to everyone from all of us here at 8 Notes. Let's get started today. We do have a fair bit to get through, so let's dive straight into it. Before I get started, of course, I'd quickly like to introduce you to the resources that we have to offer here today, and our mission statement to help you achieve the very best that you can. So you'll find that we have lots of free resources available today that you can access at our website, and they include study notes, so free study notes that are posted by your peers, by past tutors, and it's a really good way to consolidate your notes and add on to them. We of course have our lectures going on this week, so please try and come to and sign up for as many as that are relevant to you, and I'm sure you'll find them super helpful. Along with that, we have our discussion forums as well. It's very a wonderful space for everyone to sort of come together and discuss their HSC lives. Along with that, we've got videos, newsletters, ADA calculators that will come into use, I guess, later down the track for you this year, and also articles that discuss study strategies and tips, and lots more that you can check out on our website. So go to adanotes.com in order to access that. Along with that, I will be talking about other resources that we offer in order to give you that extra bit of help if you are looking, if you want it, if you need it. And coming from someone who has utilized these resources, I found them so helpful. So we've got TuteSmart, which is our own online tutoring facility run by Year 12 elite grads. And along with that, we also have our study guides and Ed Unlimited, which is like a Netflix for all our study guides. And like I said, having used these resources, I can assure you that they are so helpful. So definitely check them out if you are looking for that bit of extra help, that extra push to help you this year. And of course, before I get into it, I'd like to extend a special thank you to our sponsors who have helped us organize these lectures, including La Trobe University, RMIT, Deakin, UTS, and Macquarie. So thank you to these guys for helping us organize these lectures free for you. So let's get started with today's content. Like I said, we do have plenty to get through. We will be, and I'll get to it in just one sec, we will be covering Module 6 and 7 today. So fair bit for us to sort of work through, but hopefully by the end of this lecture you're feeling a lot more confident with the topics, and I hope you feel like you have a fundamental grasp on these ideas that you can hopefully build on later when you go through this content in class, or when you go about consolidating your notes. So let's get started. Now of course, before I begin, I'd like to introduce myself. So my name is Aditi, and I graduated in 2021 as Dux with an ATA of 95. So I've got band 6s in English Advanced, Extension, Biology, and PHPE. I've also done Chemistry and Maths 3 units. Along with that, I am currently in my second year of Bachelor of Science and Bachelor of Advanced Studies Medical Science at the University of Sydney as a Deluyol Scholar. So I'm majoring in Medical Science and Political Economy. Other than that, huge Potterhead, love reading. That's my English side, love reading. And if you have any questions about any subjects, any university related questions, please feel free to ask, and I'll be answering them as we go. And so yeah, feel free to ask, of course, as many questions as you like about the content that we're going through today, and in general about biology, about HSC. I'm more than happy to answer anything. So today's agenda, like I said, is for us to recap Module 6. Then we will go through Module 7. So help you get a foundational knowledge for Module 7. And then we'll discuss study tips and exam strategies. Now, I'd just like to flag, I guess, as you're watching this lecture, definitely take notes. Obviously, it helps a lot with, trying to stay attentive. And again, I know, sometimes it can be so hard to sit behind a screen for 2 hours and be focused the whole entire 2 hours. Not the easiest thing to do. So definitely try to jot down things that stand out at you, ask lots of different questions, and I'm always happy to go ahead and, explain things in different ways. If one way didn't make sense, I definitely do that. So let's get started today by firstly revising our sort of big ideas from Module 6. So we won't go through obviously the whole Module 6, but we will be revising the big ideas and the key concepts so that we can then go on and spend a lot more time going through Module 7. So, as you would hopefully by now be familiar with it, this is what the HSC syllabus for biology looks like. You are studying effectively 4 topics. The first one is heredity, followed by genetic change, infectious diseases, and non-infectious disease and disorders. So, since generally most schools sort of stick in that order of Module 5 to 8, I'm assuming that most of you have done Module 5 and 6 and are now going to start 7, or like that would probably be the majority of us. If you haven't, if today is your first time having a go at both Module 6 and 7, in case the school has done 5 and 8, just try to follow through the best you can. Please ask lots of questions, and I'll try and break everything down so that we to those of you who have done it, you are building on your knowledge, and to those who haven't yet had a chance to do it, you can again start to build our foundational knowledge so you can take along with you into term 2, term 3 for year 12. So let's get started. So we're starting off by firstly looking at Module 6. So the first dot point of Module 6 is asking us to look at mutagens. What are the different types of mutagens, and what mutations do they cause? So, we look at what mutations they cause in the next slide, but that's sort of the first big point. What are mutagens? So mutagen is sorry, just gonna sneeze. Okay, it's been happening all day. I feel like I need to sneeze, and then I think about it and it just goes away. So apologies for that. But as I was saying, so a mutagen is an organism that causes mutations. A mutagen can also be known as a mutant. So there are 3 types of mutagens that we're looking at, including chemical, electromagnetic, and naturally occurring. So chemical mutagens include radioactive agents like uranium, other chemicals, and it causes structural changes. We've got metals, that including arsenic and nickel, that can affect the DNA replication process. And finally, we've got intercalating agents like ethidium bromide, which can cause frame shift mutations, and we'll have a look at what frame shift mutations are in just 1 second. There are also electromagnetic radiations. Those are like the most prominent ones. They happen, they're high energy, and they are highly penetrative. They alter DNA bonding, which causes the rearrangement of the structure, and now we know that because of the processes that are occurring in the DNA, they can have a huge impact on the genome, and as a result cause mutations. An example of an electromagnetic radiation mutant is UV light. So radiation from the sun can be super harmful in high doses, and as a result it can cause pyrimidine dimers. So that is when you would have the bases sort of, and actually there's an image here. I was trying to explain it with my hand, but I'm pretty sure I've got an image here for us. So this is the electromagnetic spectrum, and we can see that in terms of electromagnetic radiation, we would know that gamma rays, so it goes from radio to gamma, and this side as you go along it becomes more penetrative. So that's that. Okay, moving on. So now here we go. These are our pyrimidine dimers. So we've got as you can see, so what happens as a result of radiation to the DNA, so there can be hydrogen bondage breakage. So by now, as we would know that the bonds are joined, so the bases they have hydrogen bonds, holding them together. So hydrogen bonds here can break. They can also be pyrimidine dimers. So this is when you can see the bases, they turn inwards to connect to one another instead of connecting straight across. There can be a loss in bases, base change, cross linkage can also happen. There can be single strand break and also protein cross linkage. So there are lots of things that can happen here. I would say to really know pyrimidine dimers, what that is, how that happens, and of course hydrogen bond breakage. And yeah, those are sort of the big ones that we need to know because they have a huge role, in changing the structure of the DNA. Okay, moving on now to inquiry question 2. So how does mutation introduce new alleles into a population? So firstly, as you can see here, these are firstly the results of mutagens. So it can have a huge impact on phenotypic expression. And as you can see here, we've got ionizing radiation that has affected the DNA. So we've got the X-rays or gamma rays over here breaking the single strand and also breaking the double strand. On the other hand, we've got non-ionizing radiation, which causes pyrimidine dimers to form between thymine. So it can form between thymine and guanine. Yes, it's always good to remember that. But this is an example here of thymine dimers. So these are forming between two thymine bases. So that's that. All right, let's have a look now at what happens as a result of these mutations. So we've got point mutations. So point mutations are a type of mutations that happen as a result of damage to DNA, and there are two sort of distinct categories for them. The first one is substitution, so they can be substitution mutations or they can be frameshift mutations. So with frameshift mutations, there are two specific types. There is insertion or deletion. So we've basically got the three types of mutations that happen as a result of these and the three effects. So the three types are, they can be silent mutations. So silent mutations is when there is no effect on the codon, on the DNA. So that there's no effect, it's silent, it doesn't have any effect on the DNA. The second is missense, which is when we see a change in the amino acid because the codons have been disturbed. There has been a change in the codons. Finally, we've got a nonsense mutation, which means that a stop codon has been prematurely introduced, which is going to affect it is going to prematurely stop the process and affect the DNA that is being produced. Sorry, that is going to affect the proteins and the peptide synthesis that's happening. So those are firstly point mutations. Now we've got chromosomal mutations. Now chromosomal mutations is when actually let me just quickly go back just because I realized I forgot to mention something. So with point mutations, that's the big category. So there are point mutations, but there are two types of point mutations. There's substitution, when a base has been switched out, and then there's framework, when either a base has been inserted or it has been deleted. And so as a result, you get to see three different types of effects, three different types of changes, and then there's specific effects. So just wanted to flag that out. Okay, let's have a look now at chromosomal mutations. So chromosomal mutations usually occur as a result of errors that happen during meiosis. And crossing over and that could be because crossing over occurs incorrectly. So there can be chunks of DNA that was screwed up, and that can cause structural mutations to happen. We can also see sister chromatids being incorrectly separated. So during anaphase, when the chromosomes are supposed to separate, the DNA could not, it may not be separated properly, which can then again cause a number of mutations as well. Sorry, let me just check my charger's plugged in. Just sparkling. Okay, all good. Alrighty, just making sure. Yep. Okay, perfect. So let's now have a look at the effects of it. The effects are usually severe because now we've got issues within the DNA happening at a bigger level, like it's happened during meiosis, and it is going to affect the phenotypic expression, but it's also going to affect their gametes. In some cases it can affect their gametes as well, depending on what the mutation is. So there are 4 types of structural chromosomal mutations. They include deletion. So deletion is when a chunk of that chromosome has been deleted, as suggested by the name. There is inversion, when a part of that DNA has been inverted inside out. We also have translocation, when a part of the DNA has been misplaced. The chromosome has been displaced. And finally we have duplication, where a part of the chromosome has been duplicated. So those are the 4 types of structural mutations. And if we've got mutations in the number of chromosomes, which can again happen if we've seen if it's happening during meiosis and it affects the separation of the strands, there can be mutations in the number of chromosomes. And there are 2 main types. There's aneuploidy and polyploidy. So aneuploidy is when the overall number of chromosomes of the offspring differs to that of the parents. So, for example, in Down syndrome, the individual has 47 chromosomes instead of the usual 46 chromosomes, and that is because they have 3 copies of chromosome number 21. So that's aneuploidy. Then we've got polyploidy, which is when an organism contains more than 2 sets of homologous chromosomes. So, for example, triploidy is a condition in which the fetus has 3 copies of every homologous chromosome instead of 2 copies. And that is obviously going to have a severe effect, on a large number of genes. So, we've got, as I've introduced you earlier to TuteSpot, here are some examples of, for example, little guides to looking at mutations, and these and I tutor biology here at TuteSpot. I tutor biology, PDH, PE, and English Advanced, and this is one of the examples of how we like to deliver our content. Obviously, we have a very similar sort of structure. Sorry, we have a very similar structure in terms of go through the slides, but some of the resources and the way in which we present our content includes like infographics like this, which can be so helpful in visualizing what is happening, and these are here for you if you want to have a look and use them and add on to your notes as well. Okay, now let's have a look at the second big dot point, which is biotechnology. So with biotechnology, our inquiry question is, how do genetic technologies affect Earth's biodiversity? So this is something that you will come back to, that you have obviously looked at briefly in Module 5, and you will come back to in Module 7 and 8 as well. Like, you will come back to this idea of biotechnology, and we are going to go through Module 7 in a while, so you'll sort of see how that ties up with Module 6. So there are 3 different types of uses of biotechnology that we are looking at. firstly, we're looking at the use of biotechnology in health, so medical biotechnology. And examples of that include, of course, biofarms, but those are not just the only one. And the big examples, of biotech, of medical biotech is vaccines and antibiotics. Having now, gone through the pandemic, we are all very aware of vaccines and, antivirals and stuff, and how they've played a huge role in curbing the pandemic. There are also stem cell treatments that are being developed, and that have some have been developed, some are being developed, and there are changes being made, and that includes skin grafts, treatment of cancers and autoimmune diseases. We also have industrial biotech. So some examples include biocompatible materials like biopolymers derived and also produced by bacteria or plants, and as well as biodegradable plastics. Living in a world where we are currently dealing with the effects of climate change, along with obviously many other issues, biodegradable plastics will play a huge role in the way in which our world functions. So that is again something that is being constantly researched and improved. We also have the use of enzyme biotechnologies and processes like fermentation and food production that once again play a huge role in making industrial processes more efficient. Finally, we've got environmental biotechnology, and we will again be talking about all of them in a lot more depth. But I think environmental biotech is one of those that we sort of know, like we use every day, but we don't really think about that as like biotechnology, right? So we've got bioremediation, which is development of microorganisms to help clean up pollution. And we also have the use of genetically modified crops and organisms in agriculture that is being used to better suit a changing environment. And that is something, for example, BT cotton, corn, bananas, Cavendish bananas, all genetically modified. So it is something that we obviously that we interact with every day, use every day, but we may not actually consider it biotech. So here are the three different types of genetic technologies that we will be looking at in this module. You will see that in the syllabus some of them are specified, so the ones that I've highlighted in pink there for you, those are the ones that are specified in syllabus, but of course you still want to know examples of the others as well. So we've got reproductive technologies including artificial insemination, which is used in the livestock industry. We've got in vitro fertilization, so IVF, which is a fertility treatment. And finally we've got artificial pollination, which is used in agriculture. So these three are reproductive technologies. Then we've got cloning techniques. And cloning techniques include firstly whole organism cloning, so it's used widely in livestock industry. I think by now we would all know Dolly the sheep. So Dolly the sheep is an example of whole organism cloning. There are many other out there now. Another one is Injaz the camel. That was a camel in the Middle East that was cloned. We also have therapeutic cloning, which is happening in medicine, again a field that is still being researched and explored. Finally, we've got gene cloning, which is happening in both medicine and industry. So like I said, the ones in pink are the ones that are highlighted in the syllabus, so you definitely need to know those. And the ones in blue, of course, are extra examples that I still think you should know, just in case you get a question where you don't want to keep talking about one thing and you can bring in other relevant examples. And the syllabus also says including but not limited to, so it's a good idea to have some extra examples up your sleeves. And finally, we've got recombinant DNA technology. So that includes gene sequencing, which is used in medicine and research. It includes transgenesis, which is also used in agriculture and biotech, gene therapy, ELISA, and CRISPR, which is used in medicine and is a molecular tool. So here I've got an example for us. So we've gone through the content, and now let's have a go at adapting it to this specific question, which is from the 2019 paper, the application of reproductive technologies in plant and animal breeding limits genetic diversity. So to what statement is this statement correct? A question like this, to what extent you need to specify what your what your opinion is. You have to say to a significant extent this is true, or this is not true, or this is untrue to a great extent. Something like that, where you actually answering the question. And with a question like this, you need to be able to, like, there's a lot that you can talk about. So structure is crucial. It is worth 6 marks, so that's very important. So structure is crucial, and organizing your points to sort of come back to this big idea. Almost think a bit like an English essay. You want all your evidence to link back to your main thesis, to your main contention, your main argument. So kind of going through that structure. So let's not be scared. Let's now have a go at how we would structure this. So if we've got a question like this, I would obviously want to have an introduction that gives me definition of reproductive technologies and how they are being used in plant and animal breeding, and evaluation, right? So that is the part about how they're being used. Evaluate it for me, and through that, answer to what extent do you think this is true. Then in your body paragraphs, you are giving me examples and arguments to link back to your main contention and prove that, hey, you are right. And here I might talk about 2 specific technologies. So for each one, I will define what they are, describe give a brief outline of how they work, an example, and then comment on does it limit genetic diversity. So I have to give examples of reproductive technology, because that is what the question is asking me to do. The question wants to know if the application of reproductive technologies in plant and animal breeding limits genetic diversity. So I need to give examples to back my point up here. And finally, we want a conclusion that ties everything together and reinforces your evaluation. So here you link back to your body paragraphs. Again, like in English, you're signposting your themes. That is kind of what you're doing here. So that is that. Let us now move on to Module 7. There's a lot going on here. I'll just flag that out before we get started. So we'll take it nice and easy. I want you to take it nice and easy, and we'll work through this together. I will be talking through a lot of new concepts as well. So I just want you to, see if you want to jot down some sort of the big ideas, any terms and definitions that are going to be helpful. And of course, some of the processes like immunization and stuff, there's a lot going on there. So after you sort of go away from the lecture, I would definitely recommend to further research, or or for like especially that specific topic of how the immune system works. Definitely go ahead, like watch there are lots of good YouTube videos too that I'll recommend, that I find really helpful in visualizing the whole process. So let's go through this, and I'll start off by outlining what the key concepts are. So that is what I'll be doing, and I just want you to, again, jot down the key big ideas, and just try your very best to sort of follow along and ask lots and lots of questions, please. Okay, so the first dot point that we're looking at, like always, not always, because the last module had 3 dot points. This one has 4. So the first one is, what are the causes of infectious diseases? So we're looking specifically at the causes of infectious disease. And our inquiry question is, how are diseases transmitted? So how do they, go about infecting, how do they go about infecting one person to the other? So how does that happen? Now, causes of infectious diseases. Basically, we're looking at what are these things trying to kill us every second, because there's bacteria and viruses and pathogens everywhere. So we will look at the causes of different diseases, so what the types of murderous pathogens are and how do they go about trying to achieve their target. We will look at transmission. We will discuss an experiment that was done in order to sort of identify how a disease is transmitted and sort of conclude, like what causes diseases, and finally what the impact is on society. So, what is a pathogen? A pathogen may be a word that you have probably heard, or not. Yeah, probably heard now after the pandemic, but or may not have heard. So, a pathogen is any agent that causes or is capable of causing disease to the host. So so anything that causes a disease and obviously endangers the host. So the criteria for a pathogen to sort of succeed is that firstly it needs a host in order to hijack the host's cells, or in whatever way they operate. Like a virus does that by hijacking the cells of a host, while something like a macroparasite finds it finds habitat inside an individual and uses their nutrients and whatnot. So it differs based on every pathogen, but generally they need a host, and they can only cause infectious diseases that transmit from one person to the other. They cannot cause non-infectious diseases. So the terms that I'd like you to remember here are microbes. Microbes refer to microorganism. Anything that's small cannot be seen with the naked eye. All pathogens are microbes, but all microbes are not pathogens. We have lots of good microbes for example, in our gut, right? And we need them as well. So not all of them are pathogens. For example, microflora in our gut, not pathogens, play a huge role in helping us maintain good gut health. So let's have a look at the types of pathogens. You'll have to remember what they are and also examples of diseases that they cause. The way they are structured is through like incremental in terms of their sizes. So we start off with like prions, which are like 235 amino acids long. And we go all the way up to macroparasites that can be up to like 25 centimeters long. So it's sort of that progression. So prions are proteinaceous infectious particles, and they are abnormally folded protein transmits misfolded protein state to other cellular proteins. And an example of this is the mad cow disease. We then have virus, so they're non-cellular, and they're basically nucleic acid. Then you've got the nucleic acid in a protein envelope, and they replicate inside living cells. So what they do is they bind to the host cells, they gain entry into the host cells, and they've got mechanisms that help them do that. And once they're inside the host cells, they hijack processes, and in the case of COVID-19, for example, once it gains entry into the host cells through the use of enzymes in its enzymes on its surface, it gains entry into the host cell and hijacks protein synthesis in order to then control the cell. And if it does that, and I don't want to go into too much detail, but basically, if it that has caused ARDS, which is one of the leading causes of death from COVID-19. So that's like a respiratory disease. So that's how it works, and generally all viruses do that, but obviously differing based on the virus, it differs. So with SARS-CoV-19, the SARS is the virus that causes COVID-19. SARS also caused the first coronavirus that happened, I think, in 2006. And there are other also diseases that it has caused, like the MERS virus in the Middle East. Okay, so an example of that is measles, right? And of course, COVID-19. Bacteria. So bacteria is single-celled prokaryotic organism. It reproduces by binary fission. So hopefully we all know what binary fission is from Module 5. And it secretes toxins, invades cells, and forms biofilms. An example of this is Salmonella. Then we've got protozoa. So now we go into eukaryotic organisms. So single-celled, it's a eukaryotic organisms, and it basically absorbs nutrients from hosts. It secretes toxins, invades cells, and form biofilms as well. An example of a protozoa is malaria, and we will talk about malaria in like 1 second. Finally, no, actually we've got two more to go. So we've got fungi, which is eukaryotic. It can be multicellular, and it reproduces by spreading spores. An example of a disease, thrush. And finally, we've got macroparasites, which are visible to the naked eye, and they can include ectoparasites, those that live on the organism, and also endoparasites that live within the organism. So things like ticks and tapeworms are an example of that. Okay, so malaria. You'll have to investigate, and this is in the syllabus. So you'll have to investigate the transmission of a disease during an epidemic. So there are a couple to choose from, including like Ebola is another one, but you can sort of choose any, but I've chosen to go through malaria today because it's a really popular one that's really interesting to, I guess, research as well. So malaria is caused by a protozoa, and specifically it is caused by the Plasmodium protozoa. The Plasmodium protozoa is found in the gut of the Anopheles female mosquitoes, and it transmits via vector transmission. So it transmits when an infected female Anopheles mosquito goes ahead and bites a human, and from there, so once they've bitten a human, you've got a mosquito bite. The protozoa the pathogen is transferred into your body. It goes through to your lungs, and the next time another mosquito bites you, let's say another female mosquito that does not have the protozoa bites you, they can take away that protozoa. Like it circulates to them, and that is how the disease spreads. So we've got that was like a little summary. So let's have a look at what actually happens. So firstly, we've got the Plasmodium sex cells, which reproduce in the Anopheles mosquito's stomach, forming zygotes in the cysts of the stomach wall. Then these cysts burst and sporocytes travel to the salivary glands of the mosquito, and from there it is transferred to humans when they bite humans. Sporocytes then travel to the liver, sorry, not the lungs, the liver, and they enter the red blood cells and continually multiply. And they can actually cause the red blood cells to rupture as well, because they're continually multiplying within the red blood cells. And infected cells burst, which causes malarial fever. And finally, the cycle continues when the human host is bitten by a mosquito, which passes those mature Plasmodium cells back to to the mosquito. Because what happens is once you have been infected with malaria, the Plasmodium protozoa, it stays within your body, so you can't get rid of it, which is why people see like relapses of malaria, because it stays, it's hard to get rid of, but there have been treatments for it, but now the Plasmodium protozoa has become quite what's the word? It has actually become quite resistant. It has become quite resistant to the to antibiotics. No, it has become quite resistant to the treatment, and so as a result, that treatment is people still use it, but it actually is not as effective as when it was first delivered. So you can see in the diagram over there, we've got Let me get a pen. I really love using a pen. Hopefully you can see my screen okay. So we've got, okay, here we go. So we've got the transmission to the host that happens here, and then those sporozoites, those are sporozoites, they go straight to the hepatocytes, to our red blood cells, and they affect our red blood cells in the liver. From there, they do mitotic replication. So they are replicating ridiculously fast in the red blood cells, and as a result, our liver cells rupture, and those myriazytes, which are now mature, they are released. Once they're released, what happens is they undergo sexual cycle to produce gametes instead, and you can see here, so they're producing gametes within the human body. So you've got the ring, the trophocyte, schizocytes producing the gametes over here, and then it ruptures again, lets it out, and then from there, once a mosquito bites that human, it goes into the mosquitoes, and the gametes mate, they undergo meiosis, they migrate through the midgut wall, forms oocytes, and oocysts, and the sporozoites develop, and the cycle starts again. So with all of this, you can clearly see the way in which it continues to sort of circulate, and it is hard to get rid of. It is a disease that it's hard to eradicate, because firstly, we've got vector transmission by mosquitoes, and then even after you've got the disease, it remains in your body, and that is why people get relapses, malarial fevers, because it gets not reactivated, but essentially, yes, it comes back up, and as a result, the cells start multiplying again, and then they bust and they cause malarial fever and so on. And we will talk a bit about what happens when we look at prevention and stuff later on in the dot point. But you don't need to know in that much detail. You kind of just want to know what I've got here in the dot points for us. All the sort of the terms here, that's just me going into a bit extra detail, because I personally find it very interesting, and I think it's interesting to sort of understand what's happening at the root levels, but you just have to know what the main points are and how it's transmitted. Just because you can get questions which are asking you, how does a named disease transmit and affect people? Obviously it's going to be worded different, but something like that, and that's when you want to be able to name a disease and go through the steps of how it infects everyone. So now we look at an experiment that you will do in class, which aims to investigate the presence of microbes in different water sources. So basically, this is just to sort of give you a summary of what it might look like. Obviously, the results will differ based on the areas that you test and your sort of experimental design. But basically, when it comes to experiments, you want to have clear and concise aims and hypotheses, right? So aim, what are we investigating? Then hypothesis, what will happen? What will we see? Then your descriptive methods you can write out in an exam. So appropriate detail, they can ask you to write out an experiment in the exam. I'm pretty sure they did that in 2021, if I'm not wrong. So something very similar to this. You then want to look at what your expected results are. Obviously, after you've done your experiment, you would know what your results and you want to remember them. You obviously also want to talk about validity and reliability. So validity and reliability. Validity is the experiment testing the aim, and reliability is, can the results be seen as consistent. Accuracy, to what degree are the results precise, and you need to be able to apply these principles to the experiment. finally, the variables which you need to understand. So having a good understanding of the variables is important. You should be able to tell them apart. Like, especially, I think one thing that always confuses everyone is the difference between controlled variables and having a control. So an independent variable is the one that is being manipulated, while the dependent variable is the one that is being tested. So in this case, our independent variable is the water source that we are using. Our dependent variable is the microbial growth that we want to see in the type of water, so the amount and also the species. We're then looking at the controlled variables, so things that we're keeping the same. So in each sample, we have, for example, the same amount of water, the same temperature at which we are culturing those agar plates, and also doing it for the same time. Finally, controls. We can have a control can be a sealed, unopened agar plate which has nothing growing on it. That can be a control, because ultimately that will tell you that there's no bacteria grown on it because it's been unopened, it's sealed, in comparison to the agar plates that have got the sterilized water, pond water and seawater. So this might differ based on what your school decides to use. Like, if they just want to use, like, I don't know, water from a nearby lake instead of a sea, whatever works. But you'll probably have these four sets you'll have these four sets essentially. Okay, now we have a look at the modes of transmission. So you've already had, you've talked about pathogens. We have sort of been introduced to vector transmission when we've looked at malaria. We'll now have a look at the different modes of transmission, and there are essentially two main modes of transmission. There is a horizontal mode of transmission and a vertical mode of transmission. Horizontal is when it's direct control. I just like to think of it like horizontal as in proper horizontal, and just helps me think of like direct contact. It can also include indirect contact. It includes vector transmission and contamination. So these four are part of the horizontal mode of transmission. Then we've got the vertical mode of transmission, and that includes transplacental transmission, vaginal birth, and also breastfeeding. So for example, with breastfeeding, if the mother has any pathogens, like any pathogens, they can be transmitted to the child. Same thing with vaginal birth and also transplacental transmission. Okay, so now we look at two big experiments that really shaped the way in which that really changed our understanding of infectious diseases. Obviously, we now live in a time where our understanding of infectious diseases and the technology that we are using to counter them has increased and evolved significantly in a really nice way. It wasn't the same as when the initial scientists who sort of set out on this path were doing their research. So the first scientist that we're looking at is Louis Pasteur, and Louis Pasteur is he is important because he disproved the theory of spontaneous generation. So people used to think that bacteria or pathogens just spontaneously infect things. He instead said that it's not spontaneous, it's not just happening randomly out of nowhere, there's a reason for it. And he proposed the germ theory of disease, which says that all things come from pre-existing things. And to us now, that is like the most logical thing ever, right? But at the time, because they didn't have the scientific research to back that, they were just like, oh, it just happened. It can just happen. But he said, no, it is happening because it's coming from something that's pre-existing and that has contact with whatever it is that is being infected. And he shows that through his swan-necked flask experiment. So what he did is he took flasks with bent swan necks, and what happened was with the swan-necked glass, the particles in the air cannot travel through these necks without getting stuck. So if I've got something like, so firstly, let's start here. So I've got two different groups. I start off by applying heat to both groups. So by applying heat, initially what happens is I'm almost, I'm sterilizing it, I'm killing any bacteria in it by applying heat to it. I then let the flask sit for a while, but with one I let it sit while it's closed, so while it is closed, but with this one I open the flask, so I'm removing the neck and I'm letting it sit. When that happens, that means that suddenly all the microbes in the air have access to this solution right here. So as a result, all the microbes from the air are going to come in, and that is going to lead to bacteria being present in the flask. However, if I let the flask sit and I'm not removing it, I'm leaving it as it is, because of this passageway into the flask, you would see that there's no bacteria present, because bacteria cannot, come in through here, travel upwards and go down here. It gets stuck here on the side. So as a result, there's no bacteria present. And yeah, so basically apply the heat, break the neck, observe the growth. Flask with broken neck means that there's microbe growth, and flask with without the broken neck means that there's no growth. And that basically tells us that the it's not spontaneous generation. It's basically that the disease and the bacteria is coming from pre-existing microbes in the air that get access to these solutions right there. Okay, next we look at Koch's postulates. So this is where he developed a procedure for isolating and identifying microbes which cause disease. So he wanted to sort of see what is whether or not it is microbes that are causing disease. So what he did was, in all organisms with the disease, he says that, let's logically work through his steps. He said that in all animals that have a certain disease, there are going to be microorganisms, microbes present that are going to cause that disease. Those microbes can then be isolated and they can be grown in pure culture. So if I've got postulate 1 where the same microorganism, so I'm trying to prove the same microorganisms are present in every case of the disease. So the symptoms are the same. More than likely, there are going to be same microorganisms that are infecting these organisms. So here, my potential microorganism is the Anthrax bacilli. So what happens is I'm going to isolate that microorganism and grow it in a culture. So how do I isolate it? I isolate it from a dead from a tissue of a dead animal, and I make a pure culture. So I grow it in like an agar plate in my culture. Then what I do is I take the culture and I take the microorganisms from the pure culture that I've grown and I inoculate that into a healthy, susceptible animal, into another animal. Not the most ethical thing to do, but hey, that's what he did. So when we inoculate a healthy organism with a pure culture, it develops the same symptoms as the original organism. Because it's got those same symptoms, right? What that tells us is that ultimately it's that same microbe is causing that disease. However, what we do is we isolate and regrow that microorganism from that newly infected organism, and we do the process one more time. And so if same, then we realize that, hey, it's that microorganism that is causing the disease. So like I said, not the most ethical experiment, but that is how he was able to prove that microbes are causing disease. So every time you see similar symptoms, more than likely it's the same microbe. Okay, so we've already talked about this, but I'll just quickly go through this again because it is going to come up later. So direct contact, easiest way to catch the disease. You would know from COVID-19 from the pandemic. No shaking hands. We did elbows. Even fist bump was like not recommended. Always elbows. And so that is because there's that physical contact that's happening. Then we've got indirect contact, and that occurs when pathogens can live outside of host cells. So pathogens live in the environment after being spread. So something like influenza, where the pathogens are in the air, like after someone sneezes or coughs, the particles are in the air, and that can easily cause and someone can easily contract that. There's vector transmission, so insects like mosquitoes and ticks, which cause the Lyme disease, they can pass on the disease. And finally, contamination, so pathogens can be harbored in food and water, and there's abundance of nutrients because there's abundance of nutrients in food and water that encourages growth. With vector, we talked about transplacental, so the fetus can get a disease from the mother via the placenta, and examples of transplacental transmission include HIV and herpes. Vaginal birth, because there's microflora present in the mother's birthing canal, that can be passed on to the child during birth, and examples include sexually transmitted diseases. And finally, breastfeeding, so fluids and nutrients can potentially pass on infectious diseases through the milk to the child. Okay, so now we're sort of slowly starting to venture into the process of what actually happens, and we will talk about immune system very shortly, which I think most of the time people love talking about, like that's their favorite bit in module 7, and that's fair, right? Because it is a really amazing topic. So let's now start thinking about how adaptations are occurring in the host, and also that are occurring more specifically in that is more specifically occurring in both the host cells and also the pathogens that facilitate entry and transmission. So entry first, there are cell wall degrading enzymes that the pathogen will have that are able to degrade the cytoplasmic membrane of the host cell. They can also have toxins which damage host tissues, and also they can disable the host immune system. They can also have effector proteins. So effector proteins are proteins which are secreted to help pathogens to enter the cell and suppress their host's defenses. And with the case of COVID-19, effector proteins are used in order for the cell to sort of get into the host membrane through like fusion. Obviously there's so many different proteins involved, but that's one of the ways in which it happens. Also, the pathogen can be adhesive to the cell wall of to the cells of the host, and that can help them colonize the tissues. Finally, we also have extremophile pathogens, which can help survival inside the host cells. So now let's look at transmission. So firstly, protective covering, we've got bacterial capsules and viral envelopes. Reservoirs, they can allow long-term survival outside of host. So this is how they are basically these cells are basically protecting themselves while they're waiting to get into another host cell. So like I said, they've got protective coverings. They can rest in reservoirs, so through that they can survive for a while before they gain a host. They can also use vectors in some cases, which increases transmission efficiency. So that, as we saw with the Plasmodium protozoa, and the vectors can help carry the pathogen from one host to another. Structural adaptations, so injection machinery like with mosquitoes. And finally, we've got rapid species evolution, so higher rates of mutation during replication, which allows for new adaptations to arise. And we saw that with the COVID-19 pandemic, where we started off with first the coronavirus. We went from alpha to beta to gamma to the Omicron virus, right? So the way in which it replicated quite quickly and became more powerful as time and more powerful as time went by. Okay, so that is our first stop point done. Please feel free to ask any questions. Stop me, right? And I'm more than happy to go through any questions, and I'll be answering them live as we go. So hopefully, hopefully we're feeling pretty okay with that. And there was a lot happening, but hopefully we are sort of feeling confident with obviously pathogens, what they are, examples of different pathogens, and those two experiments that we talked about as well. We're now looking at the next stop point, which is what are the responses to pathogens? And our inquiry question is, how does a plant or animal respond to infection? So let's have a look. So for this question, you'll have to for this stop point, you will have to essentially memorize and be able to talk about a named Australian plant through, and you can obviously gain knowledge of that through either practical or secondary sourced investigation. More than likely you'll be doing secondary sourced information. So here is an example for us. We've got a pathogen known as the myrtle rust, and the myrtle rust affects the Myrtaceae family of plants in Australia, including eucalyptus, bottlebrush, tea trees. So you want to know an Australian plant, a named Australian plant, and there are many out there, but this is one of them. And the description of this disease, well, the myrtle rust is a fungal disease. It attacks new growth, so shoot tips and young stems. It is recognized, and the symptoms include the small purple spots, or small purple spots that have bright yellow spores. Also, it spreads by releasing spores like fungal pathogens do. It is found in New South Wales, Victoria, Queensland, and Tasmania, and the way in which the plant itself responds to this pathogen includes firstly by using its mechanical barriers like its thick cell walls, tough stems. It also uses secretory glands like eucalyptus oil, which is antimicrobial. It also has non-specific immune responses, which include production of harmful metabolites and also upregulation of antifungal proteins in order to encounter the pathogen itself. And I've got links there for you if you want to use this as an example that you can use to further research. I would definitely recommend going ahead and researching a bit more in order to have have a response ready to go. But let's move on now to having a look at so like I was talking about earlier, I'll be going through some resources again that we offer here at Tutsmar, and here is an example of our so with our service, I guess, we essentially and with my classes, like, we essentially do topic tests every term, every topic, which translates to like every term, and we also do our very own half yearlies and trials too, which happen like 3 to 4 weeks before your trials at school, so in the holidays, to help you prepare for them. So the way I like to structure my classes is that we go through the content, we try to spend an lesson, like a whole lesson, on one dot point, and then through that finish the module in 4 to 5 lessons, then spend some time revising the module, and then going ahead and doing a topic test on that specific module. And I've found that it works really well for everyone, because it helps them obviously prepare for the exams, for the assignments, and for trials and the HSC in the long term. And we of course also have resources for prelim students as well. And here is an example of those resources. Like, I love to make for example flashcards and other study resources to help students. Here is another example. This is an example of year 11 content that we have, and this is again for year 11 students, helping you organize your notes and lots of practice questions and practice papers as well. Okay, now let us move on to immunity. So now I guess this again is the sort of the meaty part of the topic. This is where we get to go into a lot of detail with the human immune system, but I just want to make sure that we are not overwhelmed by it. I will be going through this very slowly because I know sometimes I have a habit of talking fast, so I'll be slowly stepping us through this, so trust me. And of course, please ask questions as we go. Clarify anything that you have to. No question is a silly question. Something that is super important to always remember. No question is a silly question. So let's have a look. Firstly, we are looking at how does the body, that's our practice question, how does the human immune system respond to exposure to a pathogen? So there are mechanisms in our body that are in place to respond to pathogens. The first line of defense is part of the innate immunity. The innate immunity essentially has the first line of defense and the second line of defense, and it is non-specific, so it's non-specific to different pathogens. So it doesn't discriminate between I'm only going to work if it's a virus, or it doesn't discriminate between the transmission of the disease. So it's non-specific, and it is non-specific, and it's fast as well. So the first line of defense includes specifically these physical barriers, which are trying to stop the pathogen from getting in. We've got cilia, which are hair-like projections that line our airways, and they move in a wave-like motion to push pathogens away from the lung. So here, as you can see here, this is an example of cilia. And what they're doing is, because they're moving in a wave-like motion, they're trying to push the pathogens away from the lung. And a lot of the time, the pathogens get stuck in the cilia, so they don't go through to the lung. The second one that we are looking at is mucous membranes. So mucous membranes line openings, so cells lining the openings of the body, they secrete mucus, and that mucus traps the pathogens and particles. So this is again important in ensuring that the pathogen doesn't get past, it doesn't get through to your lungs. So we've got that in our noses, for example. So it's about ensuring that it does not get, so that particles that we breathe in don't make it to the lungs. Then we've got chemical barriers. So these chemical barriers include, for example, stomach acid. Stomach acid is very strong, very acidic in our guts, and it is all about ensuring, not in our guts, sorry, works obviously in that region with them, but stomach acid is super important in ensuring that any harmful bacteria, like, they don't survive those conditions, because it is because it is so acidic. There are also alkali contents of the small intestine and enzymes in our mouths that, again, if you breathe in any bacteria or any pathogens, they ensure that, again, they're very best to make sure that those pathogens don't travel down to our important organs like lungs and start infecting them. We then have secretions. So these are fluids which are secreted from the body, including sweat glands, hair follicles, and urine. So fluids secreted from these specific parts, so sweat glands, hair follicles, and urinary tract. And what they do is they aim to flush out pathogens, and they're also antimicrobial. Last but not least, the skin. The skin itself is a protective layer. If you've sort of had a chance to go learn a bit about skin, you'd notice that the skin that we have right now, it's a protective layer. What we have underneath this part is what is called the true skin, and that's the epidermis. That's the actual, skin. This is basically a protective layer for us, and it secretes, so the pores in our skin secretes antimicrobials, and the outer layer of skin is constantly shedding as well. And that is, again, so that we are getting rid of any pathogens that are on there, and obviously our cells need to grow. So the first barrier is the skin, the second barrier is the cilia, then we've got chemical barriers, and so on. So these two are related, I guess. So we've got cilia and mucous membranes, chemical barriers and secretions, and the first one is the skin. So that's the main one. Then we've got cilia, then we've got chemical barriers. So it's in the form where, this is like at the very beginning the pathogen will encounter. If it makes past the skin, it's going to encounter the cilia, and if it makes past the cilia, it's going to encounter the chemical barriers in the body. Okay, so we now look at basically the first line of defense in a nutshell is that we've got the body's mechanical barriers. Here's a pathogen trying to get in, let me in, but it's finding it hard. But obviously we know that they do tend to make it in sometimes, but most of the times the mechanical barriers do a pretty good job of protecting the body. The second line of defense is also part of the innate immunity, and the second line of defense is it includes 4 specific things. It includes the lymph system. Firstly, so the lymph system produces the white blood cells, and these white blood cells are crucial for majority of the immune response, and they're basically networks of tissue, and there are networks of tissues and organs which help to rid the body of toxins. So we've got cervical lymph nodes, thoracic duct, the thymus right there, the spleen, the pelvic lymph nodes. So all of these tissues and organs that are working together to flush out toxins and waste from the body. Secondly, we've got inflammation. So inflammation is basically swelling. So when there is the dilation of blood vessels and infiltration of inflammatory cells at the site of infection, and that causes heat, pain, redness, swelling, and acute loss of function. So there are 5 cardinal signs of inflammation: pain, heat, redness, swelling, and loss of function. That's the last one. And that generally doesn't happen. It's usually sort of this, depending on obviously what the injury is. Okay, we then have the second line of defense, which is, the third part of the second line of defense, which firstly is phagocytosis. So this is definitely a term you want to remember, phagocytosis. So phagocytosis involves special white blood cells, including macrophages and neutrophils. And what they do is, what phagocytosis includes is, these specific white blood cells change their shape to engulf a pathogen. They basically eat up a pathogen. And they do this, and then they destroy them using acid, and lysosomes play a huge role in this process, because lysosomes, as you would know, generally are responsible for the waste and disposal. So they play a huge role in actually. Firstly, we've got from here. So what we see is that the special white blood cells here, for example, a macrophage, goes ahead, it captures the pathogen right there. From there, it takes the pathogen into the body, and then we see these lysosomes come across, and they come through, and they use acid to destroy the pathogen, and through that the pathogen is destroyed, and then it is dispersed from the cell right there. So that's phagocytosis. Then we've got cell death to seal off pathogens, and this is known as apoptosis. So this involves the cells, which are this time macrophages and lymphocytes, lymphocytes, which completely surround the pathogen, and then they die. So they form a cyst, and that cyst is filled with pus, and through that they block pathogen movement and also block the nutrient supply to the pathogen, which is obviously helping the pathogen, and that causes it to die. So as you can see here, so normal, that is a regular cell, but now it's condensing. So they're basically once they've got their pathogen over here, they're basically completely, they've surrounded it, and they have, and if it is filled with pus, it's forming a cyst, and through that it is killed, and it leaves these fragments here. So this is apoptosis right there. So now we've gone through two very important ideas, the first and the second line of defense, and both of them are part of the innate immunity. With that, we will now look at adaptive immunity, but adaptive immunity is basically the immune response. The first two, obviously the immune system plays a role, but we don't actually see the key players of immune system in innate immunity. They come through in adaptive immunity. So if you were to compare with innate immunity, firstly, innate immunity is always there. Your body, those mechanical barriers, they're always there. They're not, sort of phasing in and out. They're always there. And it includes this first and second line of defense. Specificity is that it is non-specific, so it's not specific to a disease. For example, your cilia is not only going to move when it sees a specific disease. It's non-specific, and it aims to stop any disease from coming into your body. The response rate is rapid. This happens quite rapidly. Phagocytosis, apoptosis, these processes are happening rapidly. Is there immunological memory? So for example, do those cells remember, okay, this is how we have, for instance, killed these cells? No. So that means that it does not have immunological memory. And so the keywords here are always to remember innate, right? So it's innate, it is constant, it is non-specific, there's a rapid response, and most importantly, there's no immunological memory. Adaptive immunity, on the other hand, now is something that arises when the pathogen enters the body. Once the pathogen has gained entry into the body and it is not being stopped earlier on and killed, for example. So for example, in innate immunity, if it was sort of stopped in the cilia or something, or if it was stopped, let's say in the stomach acid, actually not stopped there, but it was in that region specifically, the macrophages may come and induce phagocytosis and finish off the bacteria, but that would mean that it has gained entry into the body, basically. So now adaptive immunity comes into play when the pathogen has gained entry into the body. And there are two main elements that you're always going to remember with this. Those are B cells and T cells. So B for bat, T for toy. So B cells and T cells. And this response, and these cells specifically, are highly specific. This response is specific. It is tailored to the pathogen that enters the body. So it's going to be very specific to that pathogen, because every pathogen is of course different and needs to be dealt with differently, and that is what the adaptive immunity does. The response rate is that primary exposure is slow. The first time that a pathogen enters your body, the response is going to be slow, and I'm going to explain in 1 second why that is. However, the second time that pathogen enters the body, the response is going to be a lot faster, and that is because the adaptive immunity has immunological memory. The B cells and T cells actually remember how they dealt with a specific pathogen. And so our keywords here are that it's adaptive, it is highly specific, it has immunological memory. Okay, so now we come to the really important part, which is the third line of defense. Like I said, there are two main cells that are involved. There are the B cells and the T cells. The B cells are involved in what we call the antibody-mediated immunity. So I would definitely jot this down. the B cells are involved in the antibody-mediated immunity, and that is also known as humoral immunity. So humor and then AL, humoral immunity. And what happens is we've got two types of B cells. So B cells differentiate into two types of cells. They differentiate into either plasma cells, which provide immediate protection. It secretes loads of antibodies, and it has same specificity as the parent cell. What does that mean? So let me just take one step back. When a cell, every cell has antigens on it. Antigens, and especially the antigens, is basically, you know, these sort of these signs on the surface of the cell, these little like receptors on the surface of the cell that tells the body that either these cells are foreign. So if you've got a pathogen, the body will recognize its antigens on the cell surface and go, oh, that's not from our body, that is from somewhere else. But the other cells in our bodies would have antigens that the body recognizes as self, right? Okay, this is my cell, I don't have to attack it. The immune system goes, I don't have to attack it because it is my cell. But in some cases, you know, we like if you would have heard about autoimmunological diseases, and that happens because the body is not able to recognize its own cells as like self cells, and that is why they attack those cells. So that happens with, for example, with type 1 diabetes with insulin cells, or the cells that produce insulin. But anyways, let's take one step back again. So what are antigens? Antigens are receptors on the surface of a cell that tell the body, that allow the body to recognize foreign cells versus self cells. So cells that are from your own body. That is the key here. So for those antigens that are not from your own body, your body, and specifically these plasma B cells, produce antibodies. And what those antibodies do is those antibodies go and bind to the antigen. And that is basically a way of flagging to the rest of the important cells, hey, I found this specific pathogen in the body. I've recognized that you know, it has different antigens. We've recognized that, and now we've made an antibody to it. So far, just keep that in mind. So the key words here are antigens and antibodies. So plasma cell provides immediate protection and secretes loads of antibodies to provide that immediate protection. We will talk about, you know, that's not the whole process, it's a simplified process. We will go into it, but just for now, bear with me. So that's plasma cells. The second type of cell that the B cells differentiate into is memory B cells. Memory B cells, these are the cells that remember what the antibody produced was to that specific antigen. So these cells remember, you know, that, oh, this is the antigen and I have produced this antibody to it, so I need to remember. These cells do that remembering. And so the next time, if you get that same pathogen, if it comes back, these are the cells that are going to go, oh, I already have the antibody for it, let me go deal with it. Which is why that secondary exposure to a pathogen is faster, because we already have the antibodies ready to go. So that's that. They also remain, like I said, they remain in circulation to allow for a quicker and stronger response upon reinfection. So that's the B cells. They are responsible for humoral. Let me write it down. Oh, I cannot write like that. Please excuse my handwriting. So these are humoral. Okay, there we go. Sorry, that took a lot of concentration. So yeah, so B cells are responsible for antibody-mediated immunity or humoral immunity. That is the role of the B cells. Now let us look at the T cells. I'll try and draw like a line in between here. Okay, cool. So the T cells are responsible for cell-mediated immunity. I'm going to go through a schematic of what happens in each, but just give me 1 second. I promise we'll get through it. But first, you need to know what these players are. So with B cells, there are two main players. B cells differentiate into plasma cells and memory B cells. T cells. They are responsible for cell-mediated immunity. And the way I like to remember these different types of T cells is to think of it like a friend group. Yep, so I like to think of it as a friend group. So we've got 4 types of friends. We have firstly the helper T cells. These helper T cells play a huge role in helping out the other white blood cells, including the B cells. And what they do is they secrete cytokines to help coordinate the immune response. So once they latch on to the pathogen, they secrete cytokines to then go ahead and like tell everyone, hey, there's a pathogen here, I found it, come along, we need to do the whole immune response. So I like to remember the helper T cells as like the kind, helpful friend in the group that's like really sweet. That's how I like to think of the helper T cells. So that's the helper T cells. Then we've got the cytotoxic killer T cells. I love the name. I think it's such a strong name. And these are basically the cells that cause destruction. They release cytotoxins which kill target cells by triggering apoptosis. So they see a cell, they don't think, they go ahead and they kill. That is what these cells do. That's why they're destructive. And I like to remember the cytotoxic killer T cells as like the really, the really angry friend of the group, the friend that's always like ready to go ahead and fight. I like to remember it as like that friend of the group, the cytotoxic killer T cells. It's a killer T cell. It's going to go ahead and cause destruction. Then we've got the suppressor T cell. What the suppressor T cell does, as you can see in the name, is that it controls the immune response by basically controlling and suppressing the activity of the other T cells. Most importantly, the cytotoxic killer T cell. It tells the cytotoxic killer T cell to suppress its activity. Like, mate, you can't go ahead and kill all the cells. And I like to think of the suppressor T cells as like the best friend to the cytotoxic killer T cells that always gives you that balance. Like, hey, you can't go overboard. There are some cells in the body that are our own. So you just need to focus on just dealing with the pathogens and stop. Because basically cytotoxic killer T cells, they're destructive. And so the suppressor T cell is like the best friend that calms it down and just makes sure it doesn't that the cytotoxic killer T cells don't do anything rash. Finally, we've got the memory T cells. These guys I like to think as the intellectual, like awesome, smart kids of the group. These are the ones that are like, hey, you are all, so busy in getting this whole response happening, but you forgot to actually remember what antibodies you used, how you actually went about, doing this whole response. So the memory T cells give the immunological memory in the cell-mediated immunity, and they remain in circulation again to allow for quicker response next time that there's an infection. So that's our 4 main cells. Helper T cells, the kind, helpful ones in the group that are always ready to stand up for anyone. Cytotoxic killer T cells, the friend that is very hot-headed, ready to go, ready to like fight anytime. The suppressor T cells, the best friend of that hot-headed friend that go, no mate, you need to stop, that calm that friend down. And finally, the memory T cells, the smart kids, the quiet kids of the group that are like, you need to remember what this whole process was, let me do that for you. And this just helps me like really clearly remember it. So hopefully this helped you, but if you want to find another way to remember it, sure do. I mean, the names are quite easy to sort of remember too, like helper, cytotoxic, suppressor, memory. That works as well, but whatever works best for you there. So just remember, the 2 B cells are playing a huge role in the antibody-mediated immunity or humoral immunity, and the T cells play a big role in the cell-mediated immunity. I just like to remember B cells as antibody-mediated immunity, like they're responsible for the antibody-mediated immunity. Think about B cells and antibody, like that can help body and B cells. And that's how I remember that the opposite is the cell mediated. So antibody mediated is going to be humoral. The other one with the T cells is going to be cell mediated. That is something that helps me remember it. Okay, so now let us actually have a look at what this whole process is. What is happening here? And to do that, what I might give me one sec. Okay, I'm just going to go one step ahead just because I'm going to explain this first and then go back. So what is happening now? Let's have a, actually, no, sorry. I'm going to talk about antigens and antibody first because I've mentioned it, but I haven't quite explained it properly to you yet, and we haven't seen sort of what's happening schematically, so let's do that first. So now we know that there are two important parts of the immune response. There is the cell-mediated immunity and there is the humoral immunity. So what happens is, firstly, we've got the antigen-antibody interactions. Like I said, the antigens, as you can see, are these receptors on the cell surface of the pathogen. So that is an antigen, and it has those, let me change the color of my pen so like I can actually use it. Oops. Okay, let's do green. So as you can see, it has this this triangle, you know, these shapes here, right? So it's got those shapes. That's the antigen. Now those shapes there, those and then those are the antigens that are found on the surface of the cell. Then we've got the antibodies, and there are three types of antibodies that you can see here, each for a different type of antigen space. They all have different antigen binding sites. So this is the antigen binding site, as you can see, and they're all different for the different types of antigen. And this process is called the clonal selection. So the process by which the presence of a certain antigen selects immune cells that are specific to it. So the antigen, so the antibody that is selected for the antigen is going to be specific to the antigen. And this process gives us the adaptive immunity response that is specific to the pathogen. And this is like evolution, but for B and T cells. So what happens is that we've got the antigen molecules. These are the antigen molecules, and the B cells that differ in antigen specificity. They all have different shapes, as you can see here. This one has triangle. This one has like triangle poking upwards. This is like a V shape here, and this one is like a U shape. So they're all different antigen receptors. And what happens is, sorry, these are all different antibodies. Sorry, and what happens is when they when it sees the antigen, it tries to match up. It tries to match up and find the perfect match for whatever the antigen molecule is. And whichever one fits, so in this case, this specific one fit to the antigen. And so then this cell will replicate. The plasma B cells will make lots of copies of the antibodies, and those antibodies will go ahead and they will stick to that pathogen. And what also happens is that we get lots of clones of the memory cell, so that in our body later these memory cells can circulate in case we get a reinfection. They can quickly go ahead and deal with it. And the clone of the plasma cells, these plasma cells produce the antibodies, which then come off the plasma cells, and they go ahead and latch onto the antigens wherever they find the antigens in the body. So what happens now is, I'm going to go one step ahead. What happens, actually no, this is actually, it would make more sense to go this way. Sorry, I'm like skipping ahead in my own mind, so we'll go through this first. So this is the process now, where I introduce you to a concept called the major histocompatibility complex. So every single cell, so basically present on every single cell surface, it's specifically reserved for the presentation of molecules such as antigens and identification. So there's a histocompatibility complex that is found on the cell surface of every cell, and it is used to either signal an antigen or for identification. So, so that your body is like able to distinguish between self and non-self cells. So there is two types of histocompatibility complex. The first one is present on all nucleated cells, and it presents the normal proteins. It makes the abnormal ones. So what that means is these are the normal cells. It shows the immune cells that, okay, these are the normal cells. MHC2 is only present on antigen presenting cells, and those are known as the APC. So they're only present on the antigen presenting cells. And what happens is they present the abnormal pathogenic antigens. So you can see over here that we've got in all nucleated cells, this is MH class 1, we've got the antigen binding cleft, and this is the beta-2 microglobulin, so it is part of the body, so it latches onto here. This is for all nucleated cells. But in MHC class 2, there's an antigen binding cleft, right? But here we've got the antigen itself, so beta-1, beta-2, alpha-1, and alpha-2. So what does the major histocompatibility complex do? Now I'm going to take you for a second into the world of my favorite books. If you're not very familiar with it, I will try my very best to explain it, but hopefully most of us would know what's going on. So we've got Death Eaters. The Death Eaters are looking at, in the Prisoner of Azkaban in book 3, they are looking out for Voldemort. Right, so they are looking out for Voldemort, and as a result of that, they're going to stay away from the students, but they're looking out for anyone who resembles Voldemort. And as a result of that, everyone else has to be careful not to be like near Death Eaters, because once they get to a person, like they can suck the person's soul out. That is the whole point of a Death Eater, Death Eater's kiss. And also, I want you to imagine now that the Death Eater in this case is the cytotoxic killer T cell and the natural killer cells. Like I explained to you earlier, the cytotoxic killer T cells is that destructive cell in the group, that angry young cell, which is very hot-headed. And this cell, what it's going to do is it is going to be on the lookout for antigens. If it sees an antigen on a cell surface, it is going to go ahead and kill that cell off so that it can kill the antigen. So it's like a Death Eater in the body that is going everywhere and on the lookout for a Death Eater. Sorry, and on the lookout for an antigen. And it's very dangerous. Like if it comes near a cell, just like Death Eaters, right? If it comes near a cell, like it can kill any cell essentially, but it is specifically looking for those cells with the antigens on it that are foreign to the body. Any healthy cell does not want to be killed by a Death Eater, is afraid of the Death Eater, is afraid of the cytotoxic killer cells, as Harry is, right? So let's say that healthy cells are represented through Harry, and that expecto patronum, that Patronus is is the MHC is the cell showing the normal proteins that are being produced on the MHC1 to actually show that, hey, I am not the antigen presenting cell. I'm not the one. I'm not the the pathogen. Please let me go. That is what is happening here. So let me go through it again. The Death Eater is the cytotoxic killer cell and general natural killer cells. They are on the lookout for antigens. Any normal cell, like they can go to any normal cell and they can kill it, but they won't because they're on the lookout of an antigen. That is the whole process of it. So that is why that healthy cell is going to show the normal proteins that it makes on its MHC class 1 to show the Death Eaters, to show the cytotoxic killer cells that, hey, I am a normal cell, let me go. That is what is happening. Harry does it. Harry produces a Patronus to drive the Death Eaters away, and in this case, the Patronus is the normal proteins that are being used. Once the cytotoxic killer T cells notice that, hey, it's the normal proteins, this is not the antigen-containing cell, they go away. So that's what the role of the major histocompatibility complex is. This is the analogy to help you understand it. So let us now focus on the big idea of what is actually happening. So I've talked about what happens in the cells. I have talked about basically, I've talked about what is happening. I have talked about the big idea of the individual cells. Now let us look at what happens as a whole. So let me walk you through this process. I'm going to make sure that my annotations are not messy, that you can read this. So let me walk you through this process and follow along with me. These slides are here for you. If you want to go ahead later and like write down step by step what happens, that would be really helpful, I believe. So we've got a pathogen, right? That's our pathogen. And this is the antigen that is being presented on its surface. Every cell will present an antigen on the surface. Normal cells present like the proteins they make to show that they are cells, right? Like our normal cells will show our immune systems that, hey, we belong to this body by showing us the proteins on the MHC class 1. With pathogens, they are going to show the antigens. So once the body sees this, what happens is these pathogens are immediately engulfed by macrophages. And macrophages, I cannot read that, but I believe that is, they have the MHC. So they're able to see what the antigen is here, basically. So these macrophages go ahead and they engulf the pathogen like the macrophages do. Then once they engulf the pathogen, they present the antigens on their surface. So this is where now, this is the MHC class 2 presenters, basically. These are the MHC2 class 2 presenters. So what happens is they eat the pathogen up and then they present the antigens on their surface. Right there. And then interleukins, which are like proteins in the body, they call helper T cells and B cells to the site of infections. Then macrophages, they present these antigens here, and basically we've got antibody mediated immunity here and cell body mediated immunity here. And what happens is, let me change my ink color. And what happens is with the helper T cells, so they've got the T cell receptors here, and they latch on to the antigen here, and same thing happens with the B cells. The B cell will then differentiate into plasma cells and memory B cells. The memory B cells are going to store, they're going to be stored in the lymph nodes, and the next time that this pathogen comes back, they're going to be circulating and immediately jump to action. With plasma B cells, this antibody fits the macrophage over here. Remember, this is why it takes time in that first process for the body to find the right antibody for the antigen. The process is slow because we're trying to find out which antibody, because we have lots of antibodies. We are trying to find out which one fits the antigen that is on the surface. So essentially, once it differentiates, the plasma cell clones and they let go and they secrete all of these antibodies. These antibodies then go ahead and immobilize these pathogens. They go to the pathogen and latch onto the pathogen. And what this whole process does is, now the fact that the antibodies have latched onto the antigen, the body knows that it's flagged to the body that these guys are foreign invaders. We don't want them. That is why this whole process happens, because we need to know what that antigen is that we're trying to kill. And so these antibodies latch on to those antigens. So, and once that happens, our T cells come into play. So helper T cell has gone ahead, got the antibody, right? And then it has taken the antibody, it is stored in the memory B cells. These are the smart guys like the memory B cells here, and they remember that the next time that we see this antibody, this antigen again, this is the antibody that works. We also have, we also see the helper T cells send out cytokines, like I said, and those cytokines call the killer T cells and suppressor T cells. The killer T cells comes and kills the pathogen that has the antibody attached to it, right? And it secretes perforin, which is a toxic substance to destroy the pathogen. And what happens is our suppressor T cell comes along as well, because it is the killer T cell's best friend, and it comes along and it suppresses the killer T cells, tells it to calm down once we have finished the immune response, once we have destroyed the pathogen. Because remember, it can't keep the immune response going, otherwise that becomes destructive. The killer T cells can essentially kill all the cells, and they can affect all the cells. And we have obviously seen autoimmune diseases where the body doesn't recognize the self cells as self, and they go ahead and destroy those cells and don't let them function. The regulation is all messed up. So that can happen. That is why we see these cells working together, and the suppressor T cells and the suppressor T cell suppressing and regulating the activity of the killer T cell. So this whole process starts off with the pathogen, with the antigen. The macrophages come. These are our special white blood cells. They come, they display the antigen on the MHC, wait, let me write that down, MHC2 receptors, because those are antigen presenting. Right there, these yellow thingies, right? And then after that, they go ahead and they call the helper T cells and B cells. And once these cells come, they make these antibodies that can then go ahead and latch on to all the antigens, all the pathogens. You know, the pathogen has replicated by now, so go ahead, latch on to the pathogen and deal with it. That is the whole process. So I hope this was understandable, and definitely I would suggest going ahead and watching some YouTube videos, because they really help me understand the whole process. Amoeba Sisters is a good channel. They make YouTube videos which is like generally at our HSC level. Also Crash Course, of course, and they make awesome videos as well. Like the visualization is really good in both of those videos, and if there are any other good ones that you can find out there that don't go into too much detail, but like this is the kind of detail you need to know. Moving on. Now, if you have any questions about that process, please feel free to ask. So now we look at that last big dot point of prevention, treatment, and control. The inquiry question here is, how can the spread of infectious diseases be controlled? So we have now seen, firstly, what those things are that cause the disease. Then we've talked about diseases in plants and animals. Then we've talked about specifically the human immune system. And now we talk about how we actually go ahead and control these diseases. So there are 3 levels at which we need to prevent and control a disease. The first one is local, right? So think about the small scale things that we can do that we have experienced day to day. There's immunization, herd immunity, right? So and I'll be using COVID-19 as a huge example here. So we've got herd immunity. So during COVID-19, you would have heard that term a lot. No pun intended. So you would have heard that term a lot, especially like in Britain, where they were really aiming to achieve herd immunity. Obviously, it didn't end up working out. But that is something that they aim for. So that's the herd immunity. Then personal hygiene. So things like washing hands, covering cups, cleaning surface. We've got safe health practices, so like staying home when you are sick, using contraceptives, and finally public health and information. So this is about creating awareness in society. So that's that. Then we've got regional, state, and so on a regional level, we are looking at a state and a country level. What are we doing to stop the spread of the infection? Again, monitoring environmental consider, like sanitation facilities, food and water supplies, combating climate. So with things like, for example, again, with malaria, for example, right? A huge issue in Africa, in Southeast Asia, a huge condition that is affecting a lot of people. And so with something like that, like monitoring environmental conditions, like trying to drain out swamps is a big a big strategy that a lot of these areas apply in order to stop the infection of to stop the spread of malaria, because that is the environment in which mosquitoes thrive, and that uh, and the disease is spreaded. So there's monitoring environmental conditions, improving swift identification, so surveillance, recognition, and diagnosis. This again we saw with COVID-19. There were test areas set up everywhere. There were like specific areas were being surveyed, were being surveyed and surveillanced. Sorry. Also, like something that they started to do towards the end of the, not the end, but like mid towards the end of the pandemic, was when they started to test the sewage water of specific areas in order to identify which buildings or which areas had infections. So that was another thing that they were doing, and generally creating appropriate responses. Global is when we're now looking at generally the biggest scale, and we're firstly looking at quarantine, so isolating, banning individuals from traveling. So as you would know, travel basically shut down in 2020 as a result of this in most countries. Communication is done through reputable sources, like World Health Organization, and monitoring affected and potentially affected individuals, like travel cards, documents used to track this data. For example, if you would see that during the time that there was Ebola in like 2016, it was a huge, obviously a huge epidemic at the time, and what they did was they, if you traveled to Africa, and you were coming back, they would ask you, and at the airport, anyone who was coming into Australia, they would be asked whether or not you traveled to Africa in the past week or something. I can't remember how long it was, but it definitely, they would definitely ask you whether or not you've traveled to that part of the world, because of the fact that you may potentially be carrying the disease. Prevention and control. Obviously, hygiene practices play a huge role. Quarantining, vaccination, public health programs, pesticides, and genetic engineering. So genetic engineering, novel, engineering resistance to pathogens like plants, animals, vectors, humans. You would see that there has been some scientific research and development surrounding having mosquitoes, engineering mosquitoes that don't have that can not have, or rather that are resistant to the Plasmodium protozoa, and they have red eyes. There had been some research into it recently. I can't remember when that was, but there was research into it, and there's research generally into stopping mosquitoes from carrying the Plasmodium virus. There has been scientific research into that too, so engineering mosquitoes. And gene drive, so push inheritance of modified gene in natural population. So vaccination, attenuated dead, so you can see the graph, attenuated dead or synthetic viral particle introduced to trigger a primary response. So through this, the antibody concentration, as you can see, after the first vaccine, so first vaccine on infection happens here. The primary response, the first time around the whole immune system response happens, we get, this is the antibody concentration, happens over 40 days. Happens over 20-25 days. The second time that you get it, the response, you can see the slope here is so steep, the response is rapid, and the antibody concentration is so huge the next time. So vaccines play a huge role in that, and we've seen in the pandemic that again they've played a significant role in reducing the severity of the infections and also preventing infection. Okay, now let's have a look at prevention and control, and now focusing on antibiotics and antivirals. So antibiotics are medications which inhibit bacterial cell processes to stop the growth of the bacteria or kill the pathogen. And what they do is these interfere with cell wall synthesis, they interfere with nucleic acid synthesis, protein synthesis, and cell membrane permeability. And their effectiveness is depends obviously on the antibiotic and also the disease, but we have seen antibiotic resistance become a big issue recently. And the same thing happened with the antibiotics for for malaria as well, like people that the not people the pathogen has become resistant to it. So with antibiotic resistance, the development of bacterial strains which do not respond to use of antibiotics, like there is bacterial strains that then adapt to the antibiotic and stop responding to it. That is what happened with malaria, and this arises due to a misuse and overuse of antibiotics. So you would see that if you ever have like a sore throat or a cough or whatever, you would not be given an antibiotic unless it gets like really bad. So that is an example of that. Antivirals, these are medication used to treat viral infections. So remember, based on the viral life cycle, they block entry into the cell, they block viral synthesis by inhibiting transcription, and they block release of viruses. So effectiveness, well, they are difficult to manufacture, molecules which block viruses without disrupting normal cell function. There is rapid viral evolution. There is, of course, antiviral resistance, and these are very expensive. So when we look at prevention and control, we want to think about incidence, prevalence, and mortality, three important terms. So incidence is the frequency of new cases over a specified period of time. So with COVID-19, the incidence was, number of new cases every week or every day. Prevalence is the proportion of a particular population affected by a disease. So the prevalence of COVID-19 would be everyone who's had the disease, has had COVID-19 ever since like the beginning of the pandemic, for example. And mortality is the number of deaths within a particular population as a result of it. So COVID-19 mortalities differ worldwide, but we look at COVID-19 mortalities worldwide, globally, we look at it nationally, and also based on like different states as well. Here is some info on the historical sort of onto the historical pathway of looking at infectious diseases and information surrounding infectious diseases. So we start off with Hippocrates, the very, very famous, known as the sort of the god of modern science. So Hippocrates proposes primitive form of epidemiology, noting diseases only occur in certain places because of local conditions, and then that is then obviously developed upon from the 460 BCE all the way to 1965, when the World Health Organization implements the first global epidemiological surveillance unit to predict and track the spread of communicable diseases globally. And this is just this obviously table does not go into a lot of detail. I would definitely recommend for you to research a bit more, but it gives you a good idea of that development over time, and you can definitely add on. There's so much more going on, you can add on to it. Currently, in terms of surveillance, there's event-based surveillance and reporting by healthcare professional, news reports, social media, the Global Public Health Intelligence Network, and they aim to provide real-time data on local disease activity. Web-based, of course, we have got lots of resources online that allow us to track diseases. With Our World in Data, for example, is a really good website that does that. And there was also one that became really famous that was I guess programmed by a high school student that showed the worldwide statistics for COVID-19. Modeling is used to predict how infectious diseases may spread. We saw a lot of this in COVID-19, where they actually showed models on what would happen if vaccinations weren't rolled out in a way, or what would happen if masks weren't made mandatory. So we've seen a lot of that. Looking now at bush medicine. So bush medicine considers traditional skills and practices which were used by the Indigenous Australians to maintain their level of health, and they use native flora and fauna, and also preventative and diagnostic treatments in order to treat diseases, and they also focused on the treatment of mental illnesses. So considering a holistic view of health is important, and appreciating the cultural knowledge and traditional knowledge systems that have existed long before modern medicine has come in is also very important. So national Aboriginal health strategy is not just physical well-being of the individual. So they have outlined that not just the physical well-being of the individual, but the social, emotional, and cultural well-being of the whole community. This is the whole of life view, and it also includes the cyclical concept of life, death, life. Indigenous Australian communities are diverse and distinct, which means that bush medicine is a collective term, but there's no single set of Aboriginal medicines and remedies. It is a huge collection of remedies and treatments. So finally, prevention and control, looking at firstly a patent, and patent is being used, patents obviously used, when, for example, the vaccine formula and stuff that has been patented. Different companies have patented their different molecule, or like the different processes, but generally a patent is a legally enforceable right for a device, substance, method, or process, so IP Australia. And why do we have them? Because it means exclusivity, and so your intellectual property cannot be copied. And so IP and Indigenous culture, how does that play into bush medicine and Indigenous medicine, as we've talked about? IP rights, they give people the right to use their property as they choose. The good thing here is that it's helping people to profit from the ideas. If someone else uses your idea, they have to pay you for it. Bad is that it leads to a creation of monopolies and increasing price of important creations. So this happens a lot in the pharmaceutical industry, where they patent specific drugs and antivirals and stuff, and they make money from that patent because no one else can produce that. Only they can produce, those popular drugs. And there was even an example of a specific drug that was patented by a big big pharma company, and really successful. And what happened is, after its patent expired, I think after like 4 years, they changed a molecule or something in the drug and then applied for a patent again, and they got it, and they made lots of money from it. So it's a huge thing. And when we talk about patent, it does not necessarily align with the Indigenous concept of ownership, so the chain of connection isn't acknowledged. Indigenous Australians have nurtured and protected flora and fauna for 65,000 plus years, and so it is an important contribution that they have given to science and medicine. A case study is the smoke bush in Western Australia. It is traditionally used and nurtured by Indigenous Australians for medicine. The US Department of Health and Human Services filed for a patent for its exclusive rights over active molecule in smoke bush. However, and that means that they actually got that patent. So the US government, the Western Australian government, and also a pharmaceutical company, they all profit from this patent, but the traditional owners who have used it and nurtured it do not get any recognition. And that is, of course, problematic. Alright, so let's finish off with some tips, exam tips and study. I'll try to be quick. So practice papers first. My key thing is do practice papers, do practice questions. There can be, if you're running out of time, I personally put the two together. So if I felt like there were notes that I needed to make, I would make that notes to a practice question. So if one of them, like example, is that example for like a named plant. So I wanted to consolidate my notes. I got a question which was like, name an Australian plant, whatever, and then I just wrote like my notes to it and developed an answer to it. Use others' answers to narrow down your answers. If you are answers that you aren't sure about, start memorizing your answers during the multiple choice, during reading time. Like, try to remember them as you're reading through it. And of course, ladies, you can use those manicured nails to your advantage. You have that one advantage. And of course, if you've got long nails in general, manicured or not, you can just still use them to like mark the answer with your nails. I mean, yeah, I don't think it personally worked for me because it would take me like a solid 10 minutes to see what I marked at. So I'd be like squinting at my paper, but obviously you can remember it too. General tips: equip yourselves with examples. Examples are the key in biology. Mark allocations are super important as well. So they let you know what the level of detail you want to go in. The structure your extended responses. So for your 8, 9, 10, even 6, 7 markers, structure is important. You want to guide your marker through your writing. You don't want to just, regurgitate everything on the paper. You need to make your arguments clearly. So there's nothing worse than making an examiner wade through a pile of alphabetic soup. So structure, think of it as a mini essay. Finally, get used to the HSC web. So you need to find your own little niche to answer the questions. So, you know, how would you go about answering a compare question versus a draw table? Versus like, you know, a juxtapose, no, not juxtapose, that's compare, justified question. Short answer responses, one's grade are equal to 4 marks is when you start using paragraph. Get used to generic questions like to describe that, outline this feature, assess this feature. Definition plus example plus explanation is generally 3 marks. If it's a 3 marker, you want to include all 3 elements. Do not be afraid to draw diagrams. I think it's not very conventional, but you can definitely draw them as long as you as long as you talk about it in your response as well. So roughly use 1 to 2 lines for every mark, but you can write more, like you're not restricted. Along with that, you make sure that you know the verbs. Your articulation needs to be good. You need to give me a point and then explain that point, and that comes through with practice, and use as many biological terms as you can. Extended responses, structure, like I said, examples. A long response should be 1 to 2.5 pages long almost, and legibility. So legibility is important, so that comes back to writing. If you are like not handwriting your answers, you're almost disadvantaging yourself, because that is what you have to do in the HSC. So handwrite your answers. So general structure could be, you know, having an intro, having obviously your 3 paragraphs. Third one can be optional, and then a conclusion to sum it up. Okay, that is all from me today. Thank you so much for coming in. I hope today all the content we went through today made sense. Please again ask any questions that you've got. Hopefully I've answered them as we go. Take this time before the exam to figure out what works for you. You know, put yourself in a place for success. You know, put good habits in place, and I am sure you guys will smash it. So thank you so much for tuning in today. It was lovely